then we'll be on our way. Awesome. Let me just go full screen here. Cool. So as it was mentioned, I'm going to be talking a little bit today about neuroimaging. First, sort of starting off with some important background in terms of like what neuroimaging is. What are some of the motivations for doing this sorts of research? What can we sort of infer about the nervous system from it? And then we're going to zero in on two specific types of neuroimaging, specifically functional magnetic resonance imaging and something called electroencephalography or EEG. Towards the end, because obviously this workshop's more sort of about learning about different fields and figuring out is this something that you want to explore or learn more about. I'm going to talk about what I think are some important skills for people trying to get into this field, and then we can sort of go from there if we have any questions. So I'm going to turn my camera off in a second just to make sure that I'm not like my Wi-Fi doesn't cut out or interrupt the video or anything along those lines. But I just wanted to say hi. So to sort of start off here, I did want to do a quick couple of polls to just understand who is joining us today. So if you could go ahead in the chat, pick one of these options. I want to know, how comfortable are you with neuroimaging research? So one might be completely new. Two is you kind of know what it is generally, like you can name certain techniques, for example. Three, you know about specific neuroimaging techniques and you can sort of like define them. So like fMRI and EEG are meaningful acronyms to you. You've done neuroimaging research or you're an expert. Great. So I'm seeing a lot of ones, twos, couple of in-betweens, 2 and 3. Yeah, this is great. So what we're going to be talking about is right at that sort of level. So I'm going to broadly talk about this idea of neuroimaging and what it means, but also dive a little bit into some of the high-level specifics about the two neuroimaging techniques I mentioned. For anyone who is 4 or 5, either here or is watching, this on YouTube, just know that, some of the details here I'll be glossing over just for the purpose of accessibility. So I do sort of want to quickly share a little bit about myself. So I am, as it was mentioned, a PhD student at Boston University, specifically in computational neuroscience. Previously I was an undergraduate at Bowdoin College where I studied 2 different majors, one being neuroscience and the other one digital and computational studies. And so it was kind of cool studying at Bowdoin because these 2 programs are relatively unique, at least relative to some of the schools around me. So our neuroscience department was sort of distributed across 6 different departments. So it wasn't its own standalone department, but we drew classes from across the college. So it was psychology, biology, chemistry, physics, computer science, and mathematics. And so you were able to sort of find the classes that were relevant to your interests, whether they were more psychological or more biological, and sort of pick a schedule that worked for what you wanted to ultimately do. With digital and computational studies, this was sort of the intersection of a little bit of data science, understanding algorithms and technology, and then taking those things and relating them to society more broadly. And so that was really interesting because not only did I sort of get a background in, particular algorithms, so for example, algorithms in social media, but we also sort of thought through, how can we improve these things to, have a benefit on users as opposed to, driving a particular mission for a company or something along those lines. And so I sort of took those lessons into neuroscience, and I'm very much interested in, how do we use neuroscience to improve people's lives? And so now at BU, I'm sort of engaging with a lot of different areas, but mostly sort of in this neuroscience and mathematics realm. I'm not going to define each of these different interests, but I just wanted to show that, even if you go into neuroscience or you go into, psychology or you go into healthcare, you can have a lot of different interests, both, on your own, but also professionally in your job. So I've had a lot of really, great experiences in a bunch of different areas. And so I just want to sort of reassure you that even though you're specializing in an area, you can still sort of have interests and engage with projects across disciplines. So I also wanted to do one quick aside before we got into neuroimaging here, but I got a couple of emails after our session on Monday about what is this idea of computational neuroscience and how does it relate to our typical conception of neuroscience or psychology. So they're not normally distinct, but typically it's a particular lens that you bring to traditional problems. So one thing that, or at least how I would define computational neuroscience, is that we'll go into a real biological system, whether it's the brain, whether it's your heart, something along those lines, and we'll sort of abstract out features from those systems. And so abstraction is actually sort of a term borrowed from philosophy, where we are looking at something that's highly complex. We'll pick particular features that we're interested in for a particular purpose, and then we'll ignore the rest. And so, an analogy of this in the real world, I might be sitting down at my desk looking at a tree, and then I might, draw a picture of that tree. So I might, draw the trunk, might draw some leaves, might shade a little bit to show the roughness of the bark and so forth. But what I've produced is an abstraction of the tree in the sense that it is not literally every detail or every part of the tree. It's not everything inside of the trunk. It's not every, root in the ground. It's just certain parts of it to sort of serve a particular purpose. And so computational neuro is sort of going into those systems, simplifying them, and then using math or computational techniques to sort of answer particular questions. And so that's relevant to neuroimaging because a lot of what we do is actually through this mathematical or computational lens, even if someone doesn't necessarily call themselves a computational neuroscientist. So I did want to give two quick examples. So this ranges the whole gamut of what neuroscience might be. So it goes from something like mathematical biology, where we're dealing with single cells or clusters of cells. In this case, I'm showing what's called a Morris-Lakar model, which is essentially a mathematical model of how action potentials work. So we're not going to go into this, obviously, but we'll essentially take a real-world phenomenon like the action potential, draw up some equations so that we're able to sort of understand the dynamics of it. And then, people will often build tools to help us dive into the analysis a little further. Because this is a talk today, I don't know how much interactive activities I'm going to do, but I did provide a link down here to a basic simulator to sort of show you how action potentials are actually generated. So you're welcome to sort of play around with that on your own if you would like. So that was sort of the small level, but it goes all the way up to these larger ideas, many of which we're going to be talking about today in something called computational cognitive science. And so this might be, measuring brain activity at the level of the whole brain, for example, through these things called neural oscillations. And we might try to, analyze them and relate them from across the brain to get a better idea of how a particular system works. So I just wanted to say that, it isn't necessarily one area, but rather it's a lens that you bring to the rest of neuroscience. So that sort of brings us into neuroimaging, which is one of those areas where we can bring that lens. And I wanted to start with, sort of a generic definition here. As anyone here probably could have guessed, neuroimaging is sort of this process of producing images of the structure activity of the brain or other parts of the nervous system. And so in this way, it's important to recognize that neuroimaging often will refer to studying the brain or imaging the brain, but really this goes across any area of the nervous system. So earlier I mentioned the heart. There are a lot of different nervous or neuroimaging techniques that we use to investigate the heart as a system that wouldn't necessarily apply very well to the brain more broadly. So we obviously are going to be talking more about, as I mentioned, two that just focus on the whole brain itself, fMRI and EEG, and sort of understand, what sorts of questions we can ask with these, and then also understand their limitations. And so, one thing that might sort of pop into your mind, at least on the basic level, is, why are there so many different techniques? And, if we're going to be talking about two of them, why did we come up with those two? So here's sort of a chart of a bunch of different neuroimaging techniques that sort of demonstrates this idea of why we need so many different types. So on the x-axis here, we have this sort of time scale that allows us to see, what a given neuroimaging technique can do on a particular time scale. So this describes how sort of nuanced or how nuanced it can be in taking certain measurements of brain activity. So further to the left of the x-axis, we have the scale of milliseconds, where a given neuroimaging technique is able to measure brain activity at that level, all the way up to the far right, which is on the level of days, which seems a little crazy, but it makes sense in the sense that many of those techniques deal with longer term biological processes, either after getting brain surgery, or we're seeing how water flows through the body and the brain. It takes much longer time on the scale of hours or days. On the y-axis, this is sort of the spatial scales. So as I mentioned, we could do everything from a cell up to the whole brain. As we go further up the y-axis here, we have sort of neuroimaging techniques that allow us to measure the whole brain or large parts of the brain with a great precision. But then if we're able to go all the way down towards, for example, the patch clamp, which is able to measure activity below the level of the neuron. So this might be, engaging with the activity found in dendrites of a neuron. So wide range here. But as I mentioned, the two that we're looking at are the sort of light blue in the top left-hand corner. It's labeled MEG and ERP here. But we're just going to call that category electroencephalography, or EEG, today. And then we're also going to talk about the sort of light orange, rectangle right next to it, called functional magnetic resonance imaging. So, before we get into it, I just want to also say, who is interested in this idea of neuroimaging, all these different techniques? Well, it's not just sort of neuroscientists per se, but a whole bunch of people. So we have our traditional natural scientists, where we have neuroscientists, physicists, biologists, and chemists who are engaging at all those different levels of the brain down to the single neuron to try to understand their dynamics. Oftentimes, these folks are engaging in really fundamental questions about the brain, but they can serve any sorts of purpose with their work. So it could be more applied or more basic. We also have our social scientists. So psychology is sometimes fit either into the natural scientists or social scientists. I don't necessarily see a huge distinction, but for our purposes here, I'm just categorizing them with social scientists. We also see sociologists here as well, who might read neuroimaging papers to get a good idea of how a particular sociological phenomena is impacting the individual at a neurological level. And this might be in terms of poverty or disease, or systems revolving around race or migration, any number of things. But these folks often will engage with the literature as well. As I already mentioned, we have our computationalists, so mathematicians and computer scientists who are developing techniques to engage with neuroimaging. Doctors and healthcare professionals broadly, who are often using these techniques to sort of diagnose particular diseases. So whether that's from identifying structural disease, so whether that's a tumor, to more functional diseases, which are obviously related to the structure of the brain, but often manifest more behaviorally, like frontal temporal dementia. Last one on the list that often is sort of avoided in our science discussions, but are deeply important, are philosophers. And so not only are these folks interested in neuroimaging and how it relates to human nature more broadly, but they also do a lot of important work in terms of how do we actually measure phenomena? What are the behaviors that we're trying to measure and nail down across populations? And how do we interpret data properly? And so there's a lot of philosophers who have a really high level of technical knowledge of neuroimaging and can do some really fascinating work that puts them in dialogue with physicists, neuroscientists, and so forth. So I also just wanted to say, you can be interested in neuroimaging and be in any one of these fields or others that I haven't mentioned. Real quick, another poll, but in the chat, could you sort of let me know what you're sort of interested in of this list, natural science, sciences, social sciences, computationalist, and you can put more than one. You don't have to choose. I didn't choose, so you're welcome to do so. Awesome, great. So seeing a lot of natural sciences, social scientists, some folks who are interested in humanities. That's really fantastic. Healthcare, natural sciences, excellent. Great, great, great. Awesome. Thanks so much. So we're seeing a lot of 1s and 2s, a couple of 4s in healthcare, a couple of humanities, but mostly towards the natural sciences. And so this talk is going to be great for you. I'd love to chat one-on-one or anything like that with anyone interested more in, 5, for example. But this is going to be focused more on 1 and 2. And, if we have time at the end, we can discuss, how it applies to healthcare. But, sort of getting into the really fundamental idea of neuroimaging, we have a definition now. We can understand why there's different types of neuroimaging. But what's the sort of research that we're doing? So I sort of hinted a second ago at 2 different areas, structural and functional, but I wanted to sort of be a little bit more specific in what I mean by those. So structural is a sort of, allowing us to do research about how the brain is physically situated in the skull. What do I mean by this, right? So it's not literally like how it's floating in your skull, but more of, if someone has, a particular growth in their head. So I mentioned tumors, for example, or, they might have a disease progressing in a certain portion of their brain. That's something that's really important for us to understand structurally. And so there are scans, for example, MRI, magnetic resonance imaging, that allows us to have a really good and clear picture of what's going on in the skull. And so this is used in both a clinical and a research context. But at where we are right now, in 2022, most of the work that we do with the brain is less of the really basic structural scans and more of how structural relates to functional effects. And so functional effects are essentially, how does the brain change in measurable activity with respect to some baseline during a behavioral task? And the baseline, might be someone who's not doing a particular behavioral task. So if we're asking someone, for example, to add numbers together, and we're trying to see, what's going on in the brain, so we might have someone sit in there and not, for example, add values together, or they might do like a reading task or something along those lines. What I'm saying measurable is that, all of these different measurements are also abstractions of the brain, like I mentioned earlier. And so there's definitely still information that we're leaving behind that we're not able to access. And so we're trying to piece together, a picture of the brain, picture of functions and things along those lines the best we can with the tools that we have. What's really exciting about today is we constantly are getting new tools every year, every couple of years to ask better and better questions. For the purposes of this talk, we're going to be mostly talking about functional, neuroimaging, which is mostly involved in a research setting. So if you are interested in structural, I'm happy to chat offline or provide any extra resources. What do we use functional neuroimaging for? As I was mentioning, it's to get an idea of sort of how the brain is engaging with a particular behavior. Neuroimaging research is highly linked between psychology, and biology. Also a little bit into sociology, but we won't talk about that at length today. But there are 2 sort of areas where people will tend to specialize, either in functional segregation or functional integration. So functional segregation is essentially, what brain regions are active during a particular task. And so this is really helpful in a clinical and research setting, because we really need to know, if we're, for example, going in and doing brain surgery, we want to sort of know where particular regions are. So, for example, if we're trying to protect speech areas, we might do a scan before to get a good idea of someone's head to understand, this is where sort of their area is localized for speech, whereas, someone who is of a different age, different gender, things along those lines, it might be in a slightly different area. There's a large amount of variation across individuals, and so it helps us sort of understand where those brain regions are. It's also important because it allows us to know, what brain regions are helping us produce a given behavior. So a more fundamental question. We've sort of moved away from just doing functional segregation research to also include, this in functional integration research. So we might sort of identify different brain regions that are relevant in a particular behavior, but then we really want to know, how are those brain regions coordinating to produce that behavior? And so this is what functional integration is. So it sort of is the answer to, how are two or more brain regions interacting during a task in a neural network? And so this is not a neural network in the computer sense, but like a literal biological neuronal network. Neural networks in computer science are sort of like an abstraction, a further abstraction from our work in neuroscience. Once we have sort of a network mapped out, we might ask questions, how does a network change over time in response to particular stimuli? And so that might be either, on the scale of, seconds to minutes, or it might be longitudinal studies where we're asking questions, how are people's brains changing over years? And so there's a wide amount of research in each of those areas. I do want to sort of dive into one technique for each of these techniques, or each of these research areas, rather, one being fMRI, as I mentioned, the other being EEG, to understand, what does this research actually look like? I'll mention this at the end, but it should be said that, fMRI and EEG can be used in both of these areas. So they're not exclusively, put in one category or the other, but they're good to sort of communicate the gist of the field. So, we might start with functional segregation just because it's the older of the two. So functional segregation again, is the idea that regions can have specialized functions, behavioral functions. And so one of the earliest sort of examples of us coming to this realization is with this famous case of Phineas Gage. So Phineas Gage is someone who's from my home state, from New Hampshire, who lived in the 1800s. I believe it was his 20s, he was working as a railroad worker in Vermont, and he was sort of laying some new track. And so to sort of do so, they had to use all sorts of pretty dangerous tools. So different levels of explosives to remove rocks. They had large metal rods, both for the actual tracks themselves, sort of the stakes that held them into the ground, as well as others for various tasks. But one day, so he was essentially laying some track and ended up getting distracted. And there was an accident, and essentially one of the metal rods that holds the track into the ground shot up through his head and sort of ruined his day. But what was really exciting from a research perspective and also from a moral perspective is that first he recovered fully. So he did not have any extensive physical damage. So on the picture, or the picture on the left, is a picture of him after the injury, after he recovered. He did unfortunately lose his eyesight, but more or less he appeared physically fine. But what was really exciting from a research standpoint is that people started to notice particular changes with him. So some of the changes that were reported are, a little exaggerated, but we did sort of see a change in his personality, his ability to sort of make certain decisions. His moral reasoning skills changed a little bit. And so after extensive behavioral research, we saw that, a lot of his intelligence and his spatial reasoning skills and things along those lines were totally fine. but it led us to sort of infer that, certain personality traits and decision-making abilities were localized to particular regions of the brain. And so this sort of set off this long race that started very slowly, but picked up a ton in the late 20th century to sort of map the brain in terms of what a given region influences behaviorally. And so shown here is a basic framework for how we might go about thinking about this. So we might pick a particular brain region. We might sort of measure the activity in that region in one way or another while someone's doing a particular task. So whether it's, a color identification task, a linguistic task, a mathematical task, whatever it might be. And we try to understand, if that brain region is necessary and sufficient for producing that behavior. Necessary in the sense that we see people who have damage to a particular region, like Phineas Gage, are unable to complete a particular task, or their performance at that task is heavily altered. So that's one area. Or we sort of see in more modern examples, higher amounts of brain activity during a particular task. So is that brain region being recruited to complete, one of these tasks in a lab or in the real world? And then we try to, theorize about what that particular brain region does. And so people will often write, long literature reviews looking at all these different studies and try to pinpoint what exactly a particular region might be doing. And so, the framework here, and this is one that we still use today, is often sort of borrowing a behavioral task from cognitive psychology. Anyone who's ever taken a psychology class might understand how difficult it is to actually define one of these tasks and make sure that it measures a particular construct consistently and reliably over time. But us in neuroscience are grateful to sort of borrow from that discipline and use those tasks to sort of get at the more biological features of the behavior of interest. So we'll then, measure the brain activity in some way. This is typically seen as cognitive neuroscience. And then we'll compare that brain activity during the task from psychology with some baseline activity. And so there are some really basic tests that we might learn in early college statistics classes or late high school that allow us to compare. But there's also really sort of fancy mathematics that allow us to compare really interesting conditions within a task. But once we sort of do that in one study, we'll try to replicate that activation during related tasks and across participants and across time. And so for us, the step that we're zeroing in with fMRI in just a second is the actual ability to measure brain activity. And so, when we're thinking about this idea of functional segregation, something that's really important to be able to successfully measure, the activity in a region is to have something called high spatial resolution. And so this is something that I mentioned earlier, where we're able to sort of see, activity in the brain more broadly. But we need to be able to see, specific, identifiable regions in the skull, or through the skull rather, to make sure that the region that we're measuring each time is the same, and that we can be sure that the activity is related to the task. So we essentially need to be able to show the difference between activation during a task and when we're not doing the task. So one thing that's really relatively new, it's one of the newest versions of neuroimaging, but also perfect for this particular task, is something called functional magnetic resonance imaging, or fMRI. And so fMRI is sort of a generalization of just MRI that looks at the functional or the structural characteristics of the brain, but functional allows us to sort of measure the brain in time while doing particular tasks. And so it does this by sort of inferring brain activity through measuring blood flow. And so that's a really important point here, because oftentimes people will talk about fMRI as sort of measuring brain activity, right, more broadly. I've been gesturing at that throughout. But it's actually not a direct measure of brain activity. It's a proxy for brain activity. And so that's really important in terms of how we interpret things, but also in terms of the types of experiments that we can do. And we'll get into that in just a second. But what is an fMRI, and more specifically the MRI that allows us to do functional scans? Well, it's essentially a huge magnet in a room. So these things are absolutely enormous. They have entire rooms dedicated to them. And when I mean that they're large magnets, an average scanner used in fMRI is often 50,000 times greater in terms of its magnetism relative to the Earth's magnetic field. So they're incredibly strong. There are old pictures of what happens when you put any sort of metal object in the room. It'll sort of fly towards the scanner, chairs, tables, etc. It's really crazy. So when we put people in there, we want to make sure, they're not having any, special sorts of metals that will cause them any problems. People many years ago who used to have particular types of tattoos also couldn't go in because certain tattoos were magnetic. That's not an issue anymore. But there were all sorts of things where the magnet was so strong that we found out that it was an issue. But how does this sort of work? Well, the basic idea is that brain regions recruited during a particular behavioral task experience increased blood flow. So the analogy here is, when we're running, you might need more oxygen than when you're stationary. And so while you're running, you are breathing heavier to oxygenate more blood, and then that blood then delivers that oxygen to whatever areas need it, usually your muscles and your brain in the case of running. And so it's the same thing in the brain. So when it's a particular brain region is active, it needs more oxygen. So increased blood flow goes there with more oxygen to complete the task. And so why is this relevant? Well, it turns out that blood is actually weakly magnetic when it's oxygenated. So here on the left is sort of a depiction of what occurs when someone is resting versus activated. We see an increased amount of oxygen in the hemoglobin, which is, in our blood. And then it's being delivered the oxygen is being delivered to our neurons. So the two little blue cells are a particular type of neuron. And so, that's really great because we're able to sort of get these really nice pictures that I'm showing you here on the screen. So we're able to see, particular regions that have typically is denoted as like a glowing red, but an increased level of activity during a particular task. But one issue with fMRI is that it has what's called low temporal resolution. So low temporal resolution in this case is that we can't really measure activity on milliseconds, right? The best we can do in this case is on the order of seconds. And that's because blood is slow relative to electrical activity. And so, as we're increasing, our repetition of a particular task or something along those lines, it takes time for blood to become oxygenated and then go up to the brain. And so that's very different than one of our techniques that we can sort of have that millisecond resolution. And so this sort of allows us to do some sorts of functional scans, particularly in localizing function to a given brain region. But we can't ask really important questions like what are the time dynamics of a particular brain region activating to complete something. We also can't answer questions of like how long it takes for something to occur. But as we'll sort of compare this to in a second, that's okay because we have a really good sense of location in the brain. And so typically people who use MRI and fMRI will ask questions related to the place, as we mentioned, instead of the time scale. Another misconception that I sort of just wanted to briefly talk about here is the difference between fMRI and MRI, not necessarily conceptually the difference between the two, but what pictures look like. So when we sort of go back to the slide before, we see this really nice brain scan and then these red blotches on certain areas that are activated. This is not actually the picture that researchers will see. Rather, we have to do MRI scans, which take these beautiful structural pictures of the brain. Another one's right here, sort of in the center, labeled MRI. But when we're talking about functional scans, they actually look kind of terrible. So the ones on the right that are all blurry, those are actually our functional scans. And so we can sort of see patterns of activation in those, and then we're able to sort of map those activations back onto the structural scans. And so typically we have to do both of these together in order to do our research. But there's a lot of physics involved. And so one thing that I'll sort of mention right now is that, MRIs are really great, but they're extraordinarily expensive, not only because they're millions of dollars to manufacture, but because they require a full-time team. So oftentimes, these are in large hospital systems that are well-funded. And you'll have, a physicist who's on staff to be able to make sure if there's anything that goes wrong with the magnet, they're able to fix it. They also help design sort of experiments and help, doctors and researchers run studies. There's also usually different technicians in the room who will assist with those study designs. Then, of course, there's the folks who, if it's, in a clinical population, are helping patients prepare for going into the MRI, or if it's in a research setting, there's folks who are, designing like the conceptual side of the study. But a whole bunch of people are required for this to work, just because a lot of work is involved to create those really beautiful pictures. As I mentioned, sort of another pro is it's got that high spatial resolution. So we can basically see the surface of the brain and into the brain at a level of 3 millimeters with functional scans, in 1 millimeter with structural scans. And so the fuzziness on the right and the clearness on the left is the difference between about 1 to 3 millimeters, which seems crazy, but as we'll sort of see in a second, it can get much worse in terms of spatial resolution. As we mentioned, it's bad for low temporal resolution. And so we might be able to clearly see where something is during a particular task, but not have a very good idea of what is the time scale of how that thing works. So we need to borrow from other techniques to be able to do that. But another pro relative to things like lesion studies, where someone is getting brain surgery and a certain area of the brain is removed, or incidental lesion studies like Phineas Gage, where someone has a terrible accident and a particular part of the brain is destroyed or removed. This is non-invasive. So, aside from the fact that we need to remove all the metal from our bodies, and folks who, might have certain metal in their body, for example, a certain pacemaker might not be able to go in. For everyone else, it's non-invasive, and you typically can't feel anything, going on. It's just maybe a little bit claustrophobic when you actually go in the scanner. So lots of pros to the fMRI, but there are some issues with it in terms of the questions we can ask. So I did want to pause here and see if we had any questions about fMRI, either in the chat, or I'm assuming you're able to unmute. And I'm happy to take like one or two now. You're also welcome to email me any questions. So I'll just pause real quick before we move into our next one. Cool. So I'm going to continue on, but you're welcome to put them in the chat, and I'm happy to answer them offline if you have any come in. But now I do want to turn to sort of this idea of electroencephalography, or EEG. This is a technique that I mostly use in my research. And so I'm going to be sort of talking about this in the context of functional integration research. So not just where brain regions are, but sort of how are certain brain regions coming together in a network, to perform a particular task. And so, typically us in EEG research rely on, or at least in my area, rely on fMRI to define really clear regions of interest during a particular task. And then we sort of go in and characterize the time dynamics. So sort of pick up where they left off. And so what's really exciting, at least to me, about EEG is that it is a direct measure of electrical activity in the cortex of the brain. So while fMRI is only measuring oxygen dynamics, this is the literal activity from neurons. You'll notice that I was very careful to say the cortex of the brain, which is the more superior outside level of the brain, sort of denoted in that cartoon picture on the left. And that's because EEG can only measure electrical activity from cells that are vertical. And it doesn't have to necessarily be vertical in the sense that it's up or down in a particular area, but just like, it could be on the side of the brain. And, if you're looking head on, it looks like it's horizontal, but they need to be in a straight line in that sense. And they all need to be organized in a homogeneous pattern. EEG is also the one of the oldest neuroimaging techniques. It's not, I don't believe it's the oldest, but we discovered it all the way back in 1929. And it's been used in a lot of research and clinical contexts since then, particularly in, sleep and also disease detection. How is this sort of measured? Well, it's also non-invasive, and we sort of put a cap on someone's head, and we have a series of electrodes that allow us to measure that, electrical activity. We'll get into in a second, like what exactly that electrical activity is, but it's actually pretty comfortable to wear these caps. So, essentially, modern caps now, we have a bunch of them in different head sizes. They fit pretty comfortably on your head, almost like a hat. And then essentially we're just going to put some gel between your scalp and the actual sensor. And then that allows us to have a good, ability to record the electrical activity from our brain through the scalp. So this is also non-invasive. But essentially EEG is measuring these things called electrical dipoles. And so I'm not going to go too in depth into this idea of a dipole here, but essentially, because these different neurons are all sort of vertically aligned, with the cell body being at the bottom, and then more vertically or dorsal, we have our axon and dendrites and things along those lines, the synapse. We're able to sort of see a difference or separation in charge between the top and the bottom of the neuron. And this is just when it fires, again, I'm not going to explain this further, but I'm happy to chat about it offline. We see a difference in charge as an action potential propagates across a cell. And so it's the summation of all of these different charge differentials that is the EEG is actually measuring. So it is the electrical activity, specifically the current of sort of this difference when they're firing across many, many different neurons. A second ago, I used the word current. Again, we're not going to go too in depth here, but current is something that we borrow from physics to essentially describe sort of the movement of particular ions in a given closed system. And so the equation for this from physics is just I equals V over R, or I being current is equal to voltage over something called resistance, which is like an intrinsic property of a neuron. And so like wires sort of powering houses or between your phone and something like in a plug, those have certain resistance. Neurons also have certain resistance. But essentially, we're measuring that current across many of these different neurons is the main takeaway there. So because it's electrical activity, we sort of see, waves that emulate what we would expect electrical activity to look like. There are, many different peaks and valleys. They kind of look like squigglies. But essentially what you're looking at here is that electrical activity recorded from the whole scalp, where each row is coming from a particular sensor that's on the scalp. And so earlier when we saw that cap with a bunch of those little sort of dots distributed across the scalp, each of those is recording the electrical activity in each one of these rows. And so the real task of EEG is, measuring this activity in relation to a given behavioral task, and then trying to analyze it in some way. So there's a bunch of different ways that we can do so. I'm just going to briefly talk about one that's actually really new. So in the sense that, it's probably been done directionally over the last 20 years. It's really picking up now in the last 5 or 10 years. But essentially, we record those different time series to try to figure out how different brain regions are connected in functional networks. And we figure out where sort of the electrical signal is coming from and where it's being sent in the brain. And so, for example, if we're looking at memory, we might see, a certain electrical signal coming from some portion of the brain, let's call it brain region X. And then we see that same signal appearing again and again consistently in some different brain region Y. In that case, we're able to do all these fancy statistics, one of them being something called Granger causality analysis, to infer that, one is sort of causing the effect in some other area. And so I'm happy to talk about this more offline, but it's really exciting because we're able to sort of build on that fMRI research to understand, not just, oh, these brain regions are, in a network during a particular behavior, but we can really nail down both the time dynamics and which one is sort of, playing what role in a sequence of events. And so, there is a lot of really complicated parts, just like fMRI, to sort of process this data, analyze it, and be able to make these inferences. But it's a really exciting field right now because there's all sorts of new algorithms and mathematics and behavioral tasks that are coming out to allow us to do really fascinating research into, really core, portions of the human experience. So whether that's personality, memory, and things along those lines. And so there are many limitations to EEG. So one of them being that, neurons need to be aligned vertically. And so in this way, EEG can't measure things deep in the brain, right? So we can't do, things in like the basal ganglia, for example. We can't do things towards the bottom of our brain either. So the temporal lobe is very difficult to sort of measure with EEG just because of where it's situated in your head. And so this is primarily used in more cortical or, towards the top of the brain to be able to analyze those sorts of behaviors caused by those regions. If you're interested in psychology, though, that's fantastic because most of those things are what are called like executive functioning. And those are more complicated behaviors like your personality, for example, decision-making tasks, memory, as I mentioned. So those are all things that you can potentially do with EEG. The other problem is that you need many neurons to fire, and so it's not particularly nuanced in how it measures things. It needs a particular task you're looking at to sort of recruit a lot of different neurons to occur. So this would not be a good example of something that's trying to get, a good idea of what's going on at like a couple of cell level, or, a really nuanced process underlying some larger, less nuanced process. So those are just some things to keep in mind. But as I mentioned, it's really great for, memory, vision, decision-making, perception, attention, things that really require us to understand a really good, level of time dynamics underlying the behavior itself. And so, as I mentioned, the pros and cons here. So when you were looking at the data, it's a bunch of squigglies, right? So we don't really have a good idea of the spatial resolution. It's just purely electrical activity that is coming from a particular sensor that's placed in a given region on the brain. And so a long time ago, the problem with that is we didn't really know, what brain region a particular dipole was coming from. We've long since solved that problem in the sense that we have all sorts of algorithms that can go from the scalp and sort of model the interaction below this, the skull to understand where a given signal is coming from. And so it keeps improving all the time, but we're in a really good space right now where we can sort of understand, at the very least, a spatial scale of 6 to 8 centimeters cubed in the brain. Another pro is that, there's high temporal resolution, as we mentioned, so we can measure things on the level of milliseconds. Another pro, it's non-invasive, so no surgery required. You can actually just set this up on your head. There's also a really exciting area of research now where people are sort of leaving the lab with EEGs and using them in a real world context. So for example, people have gone skydiving with EEG before, and people have measured what that looks like on the brain. There's all sorts of virtual reality experiments going on right now. So it's very highly portable in that sense. So very much non-invasive and very easy to move around with. Another pro is that it's relatively inexpensive, and I use the word relatively, in good humor here. It's still like thousands of dollars. So at Bowdoin College, our EEG apparatus was about $50,000 for everything included, but relative to the scale of millions, that's pretty affordable. I did, talk a little bit about EEG and fMRI from a higher, level, and I sort of pitched these drawbacks. But what's really exciting is we can actually do EEG and fMRI simultaneously, and it's constantly getting better as statistics have been improving. So more and more people are employing both of these at once to get a really good idea of both levels of, where are brain regions being activated and how do they relate to each other in time. Another really interesting thing is we can draw on other techniques together to even supplement these further. So there are some folks at Boston University, for example, who will measure EEG, fMRI, and then a third one, for example, something called PET, which uses radioactive substances to sort of understand non-harmful radioactive substances to understand the brain dynamics. So we're continuously getting better at doing this work. And then, as I mentioned earlier, just a disclaimer for anyone who is very passionate about either EEG or fMRI and wants to sort of acknowledge the nuance here. They're both used in functional integration and functional segregation. And there are ways to sort of, improve each of them in whatever area you use them. But traditionally, you'll often see EEG being more of integration research, whereas fMRI is more of a functional segregation research. So while we're coming up towards the end here, I did want to give some advice for anyone who's interested in this work, both at a high school level and a college level. What you might see here is not necessarily what you might have expected, and there's many, many other skills that you might need. But as of right now, this is sort of what I was told and have experienced to be the most competitive for either getting into a lab or being able to easily transition into neuroimaging work. So across neuroscience broadly, but especially in cognitive neuroscience, there's a lot of coding that's involved, particularly with three languages: MATLAB, R, and Python. This wasn't the case where, a couple of years ago where we needed a lot of, we had a lot of statistical packages that were just fine for getting the job done. But increasingly, to be able to successfully do this research, people need to learn how to code. And you don't need to be, a computer scientist or a software engineer or anything like that. But it is, good to, get started with some free resources online. You can send me an email after. If you have any or you want any recommendations. But I would highly recommend sort of having these skills in your tool belt. With mathematics, if it all depends on what your school can offer, and it's not a huge deal if these aren't offered at your school, but something that you might want to take is statistics if it's offered, or if another advanced math class is offered, such as calculus, definitely choose to take that. That's less of because neuroimaging requires those courses and more of it allows you to take other important math classes that are related to neuroimaging, which I'll talk about in the next slide. Probably what I think is maybe the most important one is being able to write well, particularly scientific writing. And so I'd highly encourage people to, start a journal club where you'll read academic papers or read certain, accessible, easily understandable topics related to neuroimaging or neuroscience, and just get an idea of, what research is being done, what questions are being asked. I think that's extremely helpful, especially being able to pick apart a paper. One thing that was really helpful for me when I was back in high school to improve my writing was I worked at a student newspaper at my high school. It was relatively small, and it was sort of more club-based than anything else. So people weren't spending a lot of time on it. But I really enjoyed being able to have people give me feedback on my writing and figure out how to tell a good story. I would also say, with a blog or a YouTube channel, this is a great way to sort of, figure out how to write a script, for example, or communicate an idea. And that obviously bleeds into this idea of communication. So like practicing talks and learning how to, ask for help to, better learn or understand a given subject is super important. And then finally, skills for college students, you'll notice that much of it's the same here. So the same themes, except maybe 5, which is, recommending to either volunteer or work in a lab, either with neuroimaging, or if you don't have those resources available at your school, or if there's no sort of, researchers who use these tools, any sort of psychology or biology or even chemistry lab would be really helpful. Many of these things in scientific writing or communication are basically the same as before. So being able to communicate clearly, being able to explain topics at specific, levels of difficulty, and then being able to pick apart a paper. The last thing that I'll say in terms of, a plug, particularly for coding, is that, you don't have to have neuroimaging technology to generate data either for fMRI or EEG. And so there's a whole bunch of free data sets available online. And if you're using Python or R, those are also free. And so you can practice analyzing those data sort of by finding great tutorials on YouTube or on different blogs to sort of get those skills and increase your understanding of that work. But I do appreciate you guys, coming out today and listening to this talk. This was super fun for me. I'll see if there's any questions in the chat. I am going to leave my email here. So, we're running over on time. So feel free to send your question to me, because I don't want to sort of keep everyone here after, but I'm happy to chat as much as you need afterwards. But again, thank you so much, and really appreciated this.
So thank you so much for giving this talk. It was extremely educational. And everyone, thank you for joining. If you have any questions, please feel free to email anyone. And yeah, thank you.